And we discussed today about one modification, and it's more than 140 now modification known on RNA. So there's a lot of work in these fields. If you are interested in, do not hesitate. It's a cool field. And here's, I want to thank the collaborators, particularly Yuri and Jean-Yves and also Matthias. And that's it. Thank you very much.
Dear Professor, thank you for your presentation. Now we have questions from our participants. With your permission, we can skip to the question and answers part.
Perfect.
Okay, so our first question is, have you considered pRNA pathway level in human ovarian cell line? If yes, what do you expect to find?
Can you repeat the beginning of the question? I'm sorry.
Have you considered pRNA pathway level in human ovarian cell line?
If I consider pRNA in human?
Our technical team can send the question by chat box.
Okay, cool. I'm sorry, it's cutting a little bit. Yeah, I have it. Level in human ovarian cell. Yeah. No, we didn't look in human ovarian cell line if the pRNA pathway was affected. And it's some question I came to my mind when we start to see this defect in flies and start to work with human. But the description of the fertility of the human patient is not described. So could be a way to kind of tackle these questions, but it's not known, for example, today, at least the patients that were included in the research publication, if those patients have a fertility problem. I have no idea about it. But if yes, I guess it will be extremely interesting to look carefully at the pRNA pathway, yes.
Thank you so much. Our next question is, you mentioned the decreased lifespans of Drosophila melanogaster on your slide. What could be the future aspects of epigenetic studies about aging and prolonged lifespans? Yeah,
a very, very good question. And a lot of people in the world are working on that right now. And we have a collaboration with a team which is specialized in aging in animals, including Drosophila. And our work today is to try to understand the relation between aging and small non-coding RNA pathway default, which, I was extremely fast on this part because it was not the main topics of my presentation today, but small non-coding RNA pathway, at least two of them, pRNA and siRNA pathway, are basically here to control transposable element expression. And we know now that transposable element expression during development and also during aging are not the same. The level of these transposons are not the same, including during brain morphogenesis, indeed, during neurogenesis. It's extremely important to have some things moving around, but not too much, like in the pRNA mutants. If not, your gonads will be atrophied. So we believe that, yes, those two pathways, si and pRNA, are for sure involved in aging. And we have some evidences also that this is the case in Drosophila, but the exact mechanism of action, we don't know yet. Hopefully soon.
Thank you so much. Our next question is, I am interested on nutrigenetics and wish to know more about how lactose and tryptophan amino acid affect on the DNA.
I see. So at the DNA level, we never looked. We are really RNA guys. So, but I guess it's at one point, for example, the transcription of the tRNA are affected at the DNA level, right? Then it could give some phenotype similar to some of the one I showed you. For example, if you have more tRNA produced, then and in a condition where they will fragmented, then you will have more TRF. More TRF will means probably less small RNA activities, Argonauts activities. So it could have an effect, but we didn't go into this direction for the moment.
Thank you so much. So our last question is, This is a great chance for us to meet you. Would you kindly tell us about your educational journey? We like to hear inspirational stories to find our way to success.
Sure. Thank you very much for the comment. And again, I thank you for the invitation. It was great. I'm happy to be here, and even if it's a Sunday, because it's a sunny Sunday, so it's not so bad to talk in front of the sun, including in the lab. Plus, we are in the lockdown situation, so it makes me able to go out for work, right? So I'm glad. So my journey is, yeah, I would say, to characterize myself, so I was raised in France, and I make my studies, including my PhD in France, first in Jacques Monod Institute, which is close to here, and after we moved to the Pasteur Institute in Paris to end up my PhD. And then I moved for a postdoctoral position, first in Barcelona, in Spain, where it was extremely great institute there and cool science. And I was raised as Drosophilist geneticist. And then for my first postdoc, I switched to, originally I stay in the Drosophila lab, working on histone demethylases. And then after a year, I feel I need to learn more stuff in biochemistry because I was not really trained for biochemistry. So I moved to a lab that was more a chromatin lab, Professor Shakata, at the time at Barcelona, and then he decided to go back to where he's from, Philadelphia in the US. So I stayed 3 years in Spain and then 2 years in Philadelphia. And after 2 years in the US, I came back to France for another postdoc, working again with Drosophila. The postdoc in Barcelona and Philadelphia was not in Drosophila, was human cells biochemistry. I came back to my first love, Drosophila, but still, you see, today I'm working with flies, but also with human cells, because I think this system model Drosophila, as you know, is extremely powerful for the genetics and for RNA and DNA and chromatin or epigenetic field in general. Is extremely great if you can find some information in flies and then look into more complicated organisms, including humans or mammals, to see if the stuff are conserved and make connection with disease, which is the case for the first time at the time for FTSG1. So yes, I like when you can combine different system model if you have the possibility to do it. So when I arrived back in France, I stayed 2 years as a postdoc, and then I start to ask for position, permanent position, and I was lucky enough. It's complicated like everywhere, I guess, but I was lucky enough to get it in 2013, and then in 2000, so I'm an associate professor now, so a lot of teaching, but I like it. If not, I will not be here with you in a Sunday, right, afternoon. Yes, I think it's extremely important also to teach and continue research at I hope a good level, because this field of chromatin epigenetic RNA modification is extremely competitive. So it's exciting, but yeah, you have to be fast. So when you're a professor, you need to have great collaborators to succeed, to end up with your experiment. So my main message, if you have the possibility, I know it's more complicated for the moment. So that's why you need the vaccine. And then we will be able to move a little bit more easily into the world. Because I think it's great for me, it was a great opportunity to travel in 2 different countries than mine, Spain, of course, and the US, because you learn also a different way of thinking, of doing research, and of culture also, which is, I think, in our job, extremely important. Yeah, I can continue to talk for the whole afternoon if you don't stop me. So yes, if you have other questions, I'll take it, sure.
Thank you for your informative presentation and answers. It was really enlightening and inspiring for us.
Thank you.
And also, thank you for participating in our event. It is an honor for us to listen to you. Thank you so much again. Have a nice day, Professor.
Bye-bye, and good. And I think it's the end of your winter school, right?
Yes.
Okay, so yes. I guess it was 3 days intense. So again, thank you very much, and hope to see you guys soon around. Bye-bye.
Bye-bye. We hope so too. Dear teachers and guests, the 18th International Molecular Biology and Genetics Winter School has ended with Associate Professor Clément Carré presentation. We hope we will be together again next year. Thank you so much for your participation.